Bismillahirrahmanirrahim. Assalamu alaikum everyone. Good afternoon. This is Dr. Massab Umair. I'm working as a Principal Scientific Officer and Head of Virology Department at NIH. So today I'm going to present a topic which is basically communicating scientific data to policymakers, a case study of COVID-19 and Mpox genomic surveillance briefings at NCOC, Pakistan. So my talk has three components or three parts. In the initial phase, I will basically discuss about how we can communicate scientific data to policymakers and what are the key things to consider for scientists. And in the second part, I will briefly explain how we can communicate genomic data, especially the pathogen genomic data, to policymakers or decision makers. And lastly, I will share some of my experience of presenting genomic data with policymakers. So this is a standard definition of science policy. So it basically means the rules, regulations, methods, practices, and guidelines developed and implemented by governing agencies under which scientific research is conducted. So whenever we talk about science policy, so it has two components. One is policy for science, and the second one is science for policy. So these both terminologies, they are different terminologies and they are interlinked with each other. So what basically means that policy shapes science? Basically, it means that how policy affects the way we do science. And when we say science shapes policy, so it means that the scientific data or scientific research can impact the policy. So just for an example, how policy shapes science, I would say that we do have in Pakistan, we have a National Laboratory Biosafety and Biosecurity Policy, which basically tells scientists how to safely work on pathogenic organisms. And if we look at one of its components, which is the waste management, it basically tells that any institution or a lab that has a biological material, it needs to implement waste management systems in its institution. So what it does is that this policy tells us that only the labs that have implemented waste management system that they can work on samples that have biological material. Similarly, to explain how science can influence policy, I think a very good example is the introduction of rotavirus vaccine in Pakistan's national immunization program. So before 2017, rotavirus basically causes gastroenteritis in children, especially children under 5 years of age. So before 2017, the lab data or the lab surveillance program that was carried out at NIH, it basically showed that the rotavirus was responsible for 30 to 40% of gastroenteritis in children under 5 years of age. And similarly, this program also reported different genetic changes in the virus. And so other, epidemiologists and statisticians, they also did some research on rotavirus, and they showed that introducing a rotavirus vaccine in Pakistan can help avert mortality and childhood morbidity. So based on these laboratory findings and based on these statistical findings, the rotavirus vaccine was introduced in our national immunization program in 2017. So this is how you can see that science can influence policy. So it's important for scientists to understand what are the key stakeholders or people who are the people who make policy. So if we talk this in the context of Pakistan, so we have our parliament and our government. So these are elected bodies that draft and pass laws. So we have the National Assembly, Senate, and then provincial assemblies. So then there are federal and provincial ministries. So in terms of health, so we have the National Ministry of National Health Services and Regulation at the federal level, and then we have the health departments at the provincial level. So they all are involved in developing policies regarding health. And then there are some regulatory bodies which basically regulate different organization or institutes. So they are the enforcing organization. They also make policies. And just giving some example, we have the Drug Regulatory Authority of Pakistan, and then we have Provincial Healthcare Commission. So they are authorities or bodies that regulate different activities in the country. And in cases of emergencies, we see that different task forces and committees, they are established. And one prime example is the National Command and Operation Center. So it was established during COVID-19 to basically oversee the response at the national level. So one thing I would like to highlight here is that if you see that majority of the people that are involved in policy making, they are politicians, and they do not have scientific background, and they are not subject matter experts. So they have, you can say, they are non-technical. So as a scientist, we need to understand who are the policymakers and how do we communicate our scientific data to them. Another important aspect that scientists should understand is that who are the stakeholders or who influence policies. And again, although there are a number of stakeholders, but in broad four categories, we have scientists and researchers that influence policy through providing data, scientific data. So we have NIH, and then we have different academic institutions. So scientists and researchers working in these institutes, they basically influence policy, especially the health policy in Pakistan. We also have international organizations that influence global health policies. So we have WHO, UNICEF, and Gavi. And then we have media and civil society. It's also an important stakeholder because it plays a very key role in raising public awareness, and it also impacts decision making. And lastly, we have private sector and industry. So certain pharma companies and biotech firms, they also advocate for industry-friendly regulation. So it's important for as a scientist, we understand who make policies and who are the stakeholders that influence policies. What is basically the role of scientists in policymaking, and why should scientists engage with policymakers? So this is a very good, important question that we should understand, and we should know why, as scientists, we need to engage with policymakers. So there are three main reasons why scientists, they should engage with policymakers. We can highlight the importance of our scientific findings, and we can tell our policymakers what benefit it has. So in turn, we can secure funding, and we can secure support from our public health, you can say, policymakers or decision makers. And lastly, I think it's a very key thing that scientists, they basically bridge gap between policy and science. And they help interpret complex scientific knowledge into very simple information that is understandable by policymakers. So how can scientists influence policy, without having a formal public position or holding a public office? So there are at least four major ways, and one major mode of communication is through writing. So scientists, they can write articles, they can write commentaries, and they can write different articles and communicate their scientific finding to not only policymakers but to a wider audience and you can say general population as well. So scientists, they can also influence policy through public engagement and advocacy, and this can be done through conducting media interviews, so engaging with media and sharing scientific findings with the larger population. Similarly, scientists, they can leverage digital platforms and social media. not only to communicate scientific finding, but to dispel misinformation. And lastly, and last and most important is that scientists, they can directly communicate or have a dialogue with policymakers. So I think this is one of the most, you can say, effective way of communicating with policymakers. So before meeting with policymakers, I think it's important for scientists to anticipate that there will be some challenges on one hand, and there will be definitely some opportunities as well. And the goal of scientists before they meet a policymaker is to make sure that they overcome the obstacle or the challenge and communicate the scientific finding in an effective manner to the policymakers. And at the same time, try and leverage and utilize the chances of creating opportunities for supporting their science. So, what are some of the challenges? Time constraint. We all know policymakers, they are very busy. They have limited time. So, it's important that as scientists, we communicate our findings in a very quick and a brisk manner that is very concise. So, keeping in view the time limitation, it helps. Competing priorities. So policymakers, they have different priorities, and they are basically, they look for things that have public health impact and that have public benefit. They also look at economic factors, and they also look at the public perception. So you should take all that in consideration when you are going to talk or communicate with policymakers. Similarly, they may request you to simplify your message. So you should be prepared to communicate a complex scientific knowledge or information in a very simple form, and you should be uh, knowledgeable enough to prepare such simple and concise findings. Okay. So apart from these challenges, there are a number of opportunities when a scientist meets or communicates with a policymaker, such as a scientist can influence policy. So as I mentioned previously, if you highlight that your scientific research or your scientific findings or scientific data can help public health or can solve a particular problem, you can secure funding from these policymakers. Similarly, it provides a great opportunity to collaborate and work together with government officials and decision makers whenever you are communicating with them. And if the policymakers, they are interested in your findings, they may request follow-ups. So you may get chance to meet them on regular basis. So I think as scientists, we need to understand that there are some challenges whenever we want to interact with policymakers, but there are some great opportunities to advocate for science. So here I will, talk about 3 to 4 important things that any scientist he or she should consider whenever communicating with the policymaker. So first of all, I think it's very important that as a scientist, we should know our audience. Although here I'm talking particularly about policymakers, so we know that we will be talking to a policymaker. But whenever we are going to communicate or present our findings, it's important to know who are the policymakers. So do the policymakers have the scientific background, or they are non-technical? So it really helps in communicating scientific data. And it also helps in framing your message. And what I means by framing is that although the message, your scientific message or your data, the message remains the same when you are communicating it to a policymaker. But the way you communicate it, it can differ. So communicating a scientific finding to a non-technical policymaker, although the message will remain the same, but you will try and communicate it in a simpler form. However, if you're communicating to a policymaker that is a scientist or an infectious disease expert, here again the message will remain the same, but you will try to be more specific, more scientific. So this is something that you need to take into consideration. And so areas that you need to focus when or the things that you need to take into consideration is that you need to understand the priorities of the policymakers. So, as I mentioned, that the policymakers, they already have a lot of priorities. So they are always looking for how they can impact, or how they can benefit public, and how they can take in, or they also take into consideration the economical aspect of things. So you need to take that into consideration whenever you're talking to a policymaker. uh, you need to use their language. So obviously, when a policymaker, it's a non-technical person, you need to make sure that whatever you're communicating, it's in a very simple and actionable form that is easily understandable by them. Try to make your message relevant. I think it's very important that you try and make a connection of your scientific finding with the policymaker. Because when you make a connection, it really helps policymakers to get involved, to get they become interested, and they start listening to your message. So, how you can make it relevant? I think one example is to maybe share a case story or a patient story, and a case study or a patient story, and just to make your scientific information relevant to them. And lastly, I think it's very important to be concise and persuasive. So, as I mentioned, that policymakers, they have a lot of things on their plate. So you need to make sure that you are very concise, you are very specific about your findings. Another important thing that scientists should understand is that they should avoid jargons. So jargon, basically, it's you can say a highly technical term in terms of science. So we use a lot of technical terminologies, such as you can say single nucleotide polymorphisms, or amino acid deletions, and phylogenetic analysis. So these are called jargons, and and policymakers, they normally they don't understand these technical terminologies. It's very difficult for them to understand. So whenever, as a scientist, we are communicating to them, it's important that we make sure that we avoid such jargons. However, if there is a need to use a technical or a scientific term, I think it's very important that we briefly explain it in a simple language. So just for an example, if you are talking about a particular mutation, rather than using a word mutation, you can say a change in the genome of a pathogen. Similarly, I think it's always helpful to use analogies and examples when you are communicating scientific data to policymakers. And here I will give you a very, a example how we can avoid jargons and what impact it can have on communicating a scientific information to a policymaker. So here is the line from a famous novel, A Tale of Two Cities by Charles Dickens. And this novel basically starts with these lines that it was the best of times, it was the worst of times. So jargon, you can say interpretation of these lines would be: the times were high variance and analysis of cities, and in bracket n equals to 2. So here you can see that if you don't communicate scientific knowledge or a research finding in a simple way, it can get very confusing for any policymaker. And here also, you need to understand that if a scientific information it is not properly communicated, it can be misunderstood by policymakers, and they can lose interest into that, and it can lead to an inaction, and that is what, as a scientist, we do not need. So it's whenever we are communicating with scientists, also with policymakers and with decision makers, it's important that we try and avoid using technical terminologies. Thirdly, it's very important that as scientists, we are very definitive whenever we talk to policymakers. So policymakers, they are always looking for answers in the form of yes and no. So they want to know whether we should use a mask in public or in crowded spaces or not. So we have to be, very definitive whenever we are presenting our data, that what we want to say and how it basically impacts public health. So it's always important to, avoid ambiguities, because, unspecific words, do not help scientists whenever they are communicating. It's always good to use confident language. So as an example, I can say that if there is a finding, rather than saying that this data suggests, you can say that this data shows. So it's more confident way of communicating your scientific findings, and especially when you have strong evidence, rather than saying that this data suggests, you can say that this data shows. So it will give a very strong and a confident message to the policymaker. Also, I think whenever your message is being delivered, make sure that you provide very clear, actionable recommendations to policymakers. So they are always, interesting in what actions they can take from your data. So make sure that your message has very clear, actionable recommendation. And lastly, I think it's important to make sure that while you are trying to simplify your scientific findings, you do not oversimplify things, because again, trying to oversimplify the scientific data, it may become inaccurate, or it may not deliver the exact message that you're trying to say. So just make sure that you do not oversimplify your scientific findings whenever you are talking to a policymaker. So another important thing is that whenever we, interact with policymakers, we try to convey our scientific findings in form of a story. Normally, as a scientist, we are told to share our scientific data in form of, just share the scientific part of our findings, and we are not encouraged to share our scientific data in form of a story. But if you look some of the work that has been done by psychologists, and especially the behavioral psychologists, so they have shown that whenever a new information is told in form of a story, the people who listen to it, they remember it for a longer time, and they also understand it better. So it's a really great idea to communicate your scientific finding in form of a story. And how can you tell your findings in a compelling story form? Is that always great idea to start with a real world impact of how your findings will impact, how will they impact public health or any other um, area. And similarly, try and present the problem very clearly. What's the problem? There should not be any ambiguities. And it's always great to use data to strengthen your narrative or your message. Whenever you're telling a story, it's really great to share some of the statistics or some of the data that you have generated through science. So it really supports, and it really gives a strong, you can say, background to these policymakers. And lastly, whenever you're trying to complete your story, it's great to give them a solution. It's great to end your message on a recommendation that is backed by science. So that recommendation should be actionable, and this is how you can convince, or you can engage policymakers effectively. And here I'll give you an example. So rather than saying, 1 in 4 children in Pakistan, they suffer from malnutrition, a more effective way of communicating this would be, you can say, take a name of a person or a child that is 5 year old from rural Pakistan, struggles to grow due to malnutrition. He is not alone. 1 in 4 children in Pakistan face the same challenge. Without intervention, millions like him will suffer long-term consequences. Investing in child nutrition can change lives at scale. So it's although the message is the same, but when you communicate this message in form of a story, it becomes more emotionally relevant to the policymakers. But here, let me tell you that you should always, there should be a balance between using a data or a statistical finding along with you can say just story. So you just sharing the numbers will not be helpful, because policymakers, they are already overwhelmed with data, and just sharing a story without any numbers will not also be useful. So you need to make there should be a balance where your story is backed by numbers or statistics, and that is how you can engage policymakers. You can get their emotions, or you can involve their you can say emotions. Okay, now I'll be very specific, and I'll be talking about how we can share pathogen genomics data with policymaker and why it's important. So we all know what is pathogen genomics data. But basically, it's science where we look at the genetic material of pathogen, either virus, bacteria, or fungi. And why is it important? So basically, pathogen genomics has a lot of implications. It can be used in tracking outbreaks. So we have great example of SARS-CoV-2 and now Mpox. It can also be used for guiding vaccine development. Again, I think SARS-CoV-2 is a great example. And then this pathogen genomic data can also be used to monitor antimicrobial resistance in some of the bacteria and even some of the viruses as well. And lastly, this genomic data can be used to inform public health responses. So whether including, you can say vaccine or rolling out a vaccine at a national scale, or introducing some air restrictions or travel restrictions. So all these, they are influenced by pathogen genomic data. However, communicating a pathogen genomic data to policymakers can be challenging. And there are a number of reasons for that. Firstly, the pathogen genomic data is quite complex. So when we talk about pathogen genomic data, so there are mutations, there are different technical terminologies that are associated with pathogen genomics data. So communicating such complex data to policymakers can be challenging, and we need to take that into consideration. There is uncertainty and rapid evaluation attached to pathogen genomics. We know that pathogens, they evolve very quickly, and it's important to understand that whenever a pathogen is evolving, it's difficult to predict what could happen in future. And very importantly, the policymakers, they do not invest in something that is evolving. So they hesitate in you can say investing or giving their time to something that is evolving. So we need to take that into consideration as well. Competing priorities, as I mentioned previously, so there are a number of priorities that the policymakers have, and your problem is one of those many priorities that they do have. So you need to consider the fact that they will be looking at the financial aspects of that. They will be looking at how from the public health, from people's perspective, and they will be also looking at what benefit it can provide to a population or from a public health point of view. So just that you understand priorities that policymakers may have. And lastly, I think if this genomic data it's not properly communicated to the policymakers, it can lead to misinterpretation, and it can also lead to misinformation, and which can be very detrimental for a scientist, and it can be very dangerous for, especially from a public health point of view. So it's very important that whenever we communicate pathogen genomic data to a policymaker, we are accurate about our findings, we are knowledgeable, and we make sure that the data is presented in a very simplified form so that it can be used to take a decision. Okay, so the question is how we can effectively communicate pathogen genomics data to our policymakers. So there are a number of ways how we can accomplish this. And first and foremost, I think it's very important that our scientific finding or scientific research, it should be clear, it should be very concise. That is something that is easily understandable by the policymakers. And most importantly, it gives information that can be used to take an action. So that is very important. If we are giving some information from which policymakers they cannot take an action, they will be least bothered, and they will be very least interested in your research or your scientific findings. So just keep that in mind. I think it's always great to provide context. So let's say if I'm saying that there is an X variant, it has 10 mutations. Rather than saying that, you can say that the X variant has acquired some changes which can affect the pathogenicity or vaccine escape, and which will, directly impact the public health. So this is how, bringing context when you share your findings. Another effective way of communicating scientific data is through, sharing visuals or graphs. So we know that graphs, they are a great way of communicating data. So although you are sharing a genomic data, but it's always great to use maybe pie charts or bar charts just to communicate findings, because it's very important and very easy for policymakers to understand. Rather than just maybe sharing a phylogenetic tree, it's always helpful to share maybe a heat map or a epi data. And then lastly, connecting data to policy. So until and unless we connect our data, our scientific data, to policy, I think it becomes very difficult to convince our policymaker to invest in our science. So you need to find a way, how do you connect your data to policy? So okay, and I'll just give you some examples here from my personal experience, how I have been communicating genomic surveillance of SARS-CoV-2 at National Command and Operations Center. So as I mentioned earlier, that it's important for scientists to understand what are the audience and who are the basically audience to whom we are talking to. So at the NCOC, NCOC was basically led by our Minister for Planning and Development. So he had a background of economics and finance. So you can say he was not a scientific person. Similarly, the coordinator, national coordination, was serving Lieutenant General. So he was also not subject matter experts. But at the same time, we had some experts like our health minister. He was an infectious disease expert. And similarly, our CEO and our DG, they were scientific people. But on the other side, there were some you can say people who were not technical, and they were from army. And the purpose of including those members in NCOC was basically to support logistics during the pandemic. So here, as I mentioned, knowing that what audience you are talking to really helps. So I knew from presenting genomic surveillance at NCOC that I will be presenting a data to, you can say, to policymakers that has some technical people as well and some non-technical people as well. So I have to make sure that whatever I communicate, it's not too technical and it's not neither very generic as well. So that is one thing that I learned while communicating scientific data at NCOC, that how do you communicate to policymakers. So one of the key things that I learned from that presenting genomic data to policymakers, it can be challenging. And for the one main reason that I just discussed, that the policymaker, most of the time, they are non-technical person. So they do not have a scientific background. So how do you communicate complex scientific information to these non-technical person? It can be challenging. And this is something that we should always keep in mind whenever we are going to present our genomic findings or our scientific findings to these policymakers. So here I'll give you one example of how we used genomic data to inform public health decision making at the national level. So first of all, I think it's very important that we should, as scientists, we should understand that it's really important that we should share our genomic data in real time. Until unless our data is it's in real time, I think the effectiveness of the data becomes very very little. So if a policymaker has to take an action, the data has to be in real time, or you can say it should not be quite old. So in 2021, on the left side, you can see that our lab finding was showing that the alpha variant, which is here highlighted in the red bars, it started increasing during the fourth week of January. And in February, you can see there was an exponential increase in the spread of this alpha variant. However, if you look on the right side, the AP data or the case number, the COVID, these are the national COVID case numbers. You can see that at the end of December and at the beginning of January, the cases were going down. But we knew from our lab data that the variant, the alpha variant, it was the first variant of concern. So we knew from our lab data that the variant is increasing and there might be another surge or another wave of COVID infections in Pakistan. And we shared this data in real time with our NCOC policymakers. And exactly this is what happened. You will see that in March and in April, we saw a resurgence of COVID cases in Pakistan. And this wave, which was the third wave, it was driven by the alpha variant. So what it did, when we communicated this scientific data to policymakers, what it did that, it provided time to these policymakers to take, to prepare for a possible third wave and to take actions just in case there is a third wave. And I think this is a great example how we have used the genomic surveillance data to inform our policymakers. And here, I think another thing I would like to highlight is that some of you may know that initially alpha variant it had a deletion in a spike gene at position 6970 in the spike gene. So it was basically called a spike gene target failure. So rather than communicating, and this spike gene target failure was basically you can say a proxy or a marker, which was used to report alpha variant at that time. So rather than saying a spike gene target failure, we use the terminologies alpha and non-alpha. So this is how we avoided using jargons or technical terminologies at NCOC. Just communicating that data in a very simpler form. So these are some of the genomic surveillance work that we have done at the national level. And again, here, just to highlight that we at NIH, we have been sharing these genomic surveillance finding for SARS-CoV-2 to our policymakers almost before every wave, you can say. And I think we very timely reported the emergence and the spread of alpha variant, then the Delta variant, and similarly Omicron and its subvariants. And so, this is how you can say, communicating genetic data or genomic data in a timely manner can have direct implications on taking a decision from a public health point of view. So another thing that I mentioned, that your message should be very, clear and simple. So this is one example of how we used the testing algorithms to detect different variants of concern. But you can see that this has a lot of technical information, this flowchart. And it is not recommended to use such a flowchart whenever you're communicating to your policymakers. I'll be very honest, I used a similar sort of a chart to communicate to our policymakers. And I remember that. So I remember that when I, communicated this sort of a chart to our policymaker at NCOC, I remember that our chairman, chairman of the NCOC, who, as I mentioned, was the Minister of Planning and Development. I tried to very simply explain this complex information, and I remember he said to me that no matter how simpler you try to explain it, it will be very difficult for us to understand and to get what you are trying to say. So always try to make your message very simple whenever you are talking to policymakers. There can be, a lot of challenges when we are sharing real-time genomic data to policymakers or to decision makers, and they are, influenced by sample collection. So there can be delays in sample collection, then sequencing capacity. So we may have limited resources, and data interpretation can also be challenges. So it requires expertise as well. So I will just share one more example here. I remember that during the early phases of COVID-19 pandemic at the NCOC, we were asked that how much time does it take to sequence a sample or a SARS-CoV-2 sample. So I remember one of my seniors, he said that it takes 3 days. And this question, by the way, was asked by our chairman of NCOC. So I, being the technical and the scientific person, I knew, and I had all these limitations in my mind about sample collection. Then the need to make a batch to run NGS run, and then what are the challenges in data interpretation. I immediately very tactfully, I highlighted that although it can take 3 days, but normally it requires 5 working days to sequence a sample. So it's really important that you understand that whatever you are communicating, it is realistic, because you have to report back to these policymakers, and they will be expecting that you share that data. So it's highly relevant that you keep all those challenges and limitations that you face while generating genomic data. So we have also been, sharing some of our scientific findings in form of situation reports. Mostly we have shared these findings through our NIH website, and we have not directly shared it with our policy or decision makers. But this method can also be used to communicate scientific data. But again, you can see there that this has a lot of scientific information. So you need to make sure when if you are communicating or you are sharing or disseminating this information with a policymaker, just make sure that you simplify this situation report or this information. So another example I would like to share of my personal experience of sharing, genomic data of Mpox here this time, again to NCOC, but the difference here was that this time the NCOC was basically, the mandate of NCOC was handed over to NIH. And here you can see that majority of the members of the NCOC, they were, technical people, they were scientific people. So we had, CEO of NIH, then we had the DGs, and then different heads of department. So here again, here the importance of knowing the audience it comes into play. So here, rather than I being more generic, I had to be a little bit more you can say scientific when communicating data. So because most of the audience here was scientific. So this is a older slide, but just to highlight here that genomic surveillance in terms of Mpox has a lot of importance and implications. So Mpox has two clades, and the clade one it's more, you can say, lethal and pathogenic. It has a mortality fatality rate of around 10%, whereas the clade 2 it's less severe and causes milder infections, and its mortality or case fatality is less than 1%. So genomic surveillance can play a very key role in timely identifying what clade is present, or what clade is circulating in a particular region. And so again, we have been using this data to inform our policymakers at the NCOC here at NIH. Just one thing here important to highlight here is that if you look at the tree here, there are a lot of scientific knowledge or scientific information. If you are explaining this tree to a policymaker that is non-technical, I would highly recommend, just remove all these technical terminologies such as those accession numbers, complete genomes and partial genome. Just be very specific. Maybe write the name of the country and maybe date of collection, and then the clade as well. So these are the things that you need to take into consideration when you are communicating to policymakers. So lastly, I'll try to summarize. I think the key message of my talk here is that whenever we as scientists we are communicating to policymakers, we should understand and we should know what the audience is. So if it's a non-technical person, we need to frame our message accordingly. And secondly, it's highly important that we avoid technical terminologies. And as I mentioned previously, try avoiding using jargons and technical terminologies, because it will never go through to the policymakers. The message that you are conveying, it should be simple, and it's great to use visual aids to help explain your data. And also, I think it's very important that whatever you are reporting, it's highly authentic and it's also you're sharing it in a timely manner. If you are sharing a finding that you are not sure, so it's highly important that you do not answer to a particular question, but you can go back and you can say that let me go back and check with my colleagues, or let me go back and check with the lab data, and then I will answer that. So make sure that whatever you are talking, it's highly authentic. And lastly, I think it's great to use real world examples. So, highlighting how genomics data can impact policy decision and how it can impact basically a public health. So if we use such example, just as I mentioned about the reporting our genomic data of alpha variant, I think these type of examples, they always catch the attention of policymakers. So in conclusion, I will say that the policy and scientific research, there is a clear gap between policy and scientific research, and we need to understand what are the limitations, and we need to modify or frame our message in a way that basically aligns with you can say with the priorities of our policymakers, and that is the only way we can convince and effectively communicate our findings to them. So thank you so much. This is all that I have. If there are any questions, I'll be happy to answer.
Yes, there is one question. I'm Professor Dr. Askar Javed from Multan National Medical University. We have received 2 patients of monkeypox in the last week, and they were confirmed by the PCR lab from the Lahore, and we have also sent 3rd suspected monkeypox patient specimen to the Lahore. So if we want to differentiate the clades for this monkeypox, where should we send the specimen for genome sequencing, and what is the protocol for specimen?
Oh, thank you, sir. I think it's a pertinent question. So for clade identification of Mpox, so there are basically 2 strategies. One is based on clade specific PCR, and this is basically there are some commercial solutions available, and there are some primers and probes that are also available. So you can use those, either you can synthesize those primer and probes, or you can procure those commercial kits. So you can identify the clade based on the PCR. And the second option is that you can do whole genome sequencing. At the moment, both these things, the PCR-based clade identification of Mpox and the genome sequencing is being carried out at NIH only. So if you want to know which clade is circulating, you can refer those samples to NIH. But again, from shaping your question in terms of policymakers. So here, your policymakers, they will be very interested in knowing what clade have you found in your samples. So I think I would suggest one of the good idea would be, first of all, quickly send the sample to a reference lab. So in this case, NIH is the reference lab. And secondly, for us, I think what we will try is we basically first process it on PCR just to save time, because as I mentioned previously, whenever you're communicating genomic data to a policymaker, make sure that you share it in a timely manner. And because sequencing, it takes time, so initially we process samples on PCR to see the clade. If we don't get a conclusive answer based on PCR, then we go for sequencing.
So we have received the reports from the PCR, and the suspected monkeypox was confirmed by the PCR lab.
Yes, sir. Talking particularly about those two samples, so we do have these. We have received
Yes, they were positive. And if we want to check the clades of these two patients, should we? Which sample should we send to the NIH? And who is the focal person for this differentiation in NIH?
So at NIH, we have the Department of Virology, and the head of virology department, basically, it's me. So I am the focal person. You can send the samples to the Department of Virology. And, regarding the sample type, so it's basically, getting the swab from a lesion and putting those, swabs in a VTM. That is the sample type that is recommended for sequencing. And I think, if I'm not wrong, these samples are already received by our lab, and these samples, they are under process. My team is also online, if I'm not wrong. So these samples are already under process. So we'll share the sequencing results with you very soon, inshallah.
Okay, thank you, sir. Thank you. Maybe from the Lahore, they have been referred to the NIH for genome sequencing, because we have sent the samples to the Lahore, and Lahore has sent for the genome sequencing to your lab.
Yes, exactly. Thank you, sir. Are there any other questions?
Yes, sir. I'm Massab Umair from University of Agriculture, Massab. I'm third year student. My question is, you said there is a gap between the policymakers and the researchers in every field. What should we play our role to reduce this gap? Is not it we should have a specific seats of researchers in parliament?
I'm not sure I got your question, because there is some distortion. You mean that how we can bridge the gap between science and policy, if I'm not wrong?
Yes, sir. Of course.
Yeah. So I think here, I think one of the key thing is that as scientists, first of all, we should, and my whole talk was on this, that how we can effectively communicate our data to policymakers, and we should be only focusing on how we effectively communicate our data to policymakers. And if we do not develop these skills, no matter how great your scientific findings are, but if you do not develop the skills of communicating these data to policymakers, they will never realize the importance of this data. So we do a lot of great science, and I know that a lot of people, they are doing great scientific research, but unfortunately we lack the skills of how we can interact and communicate these findings. And also we are, you can say, hesitant or reluctant in engaging with these policymakers. So I think making sure that we have a direct, you can say, a communication channel, or we regularly communicate with policymakers, and also we develop our skills of communicating this scientific data to policymakers. I think this can really bridge that gap that you're talking about. I hope that I have answered and you understand what I mean.
Yes.
So, okay, if there are no other questions, I'm really grateful to all of you for taking time out and joining this session. So this will be, as I mentioned, this session was recorded, and this will be, available. Recording of this session will be available on our YouTube channel, Viral Insights. And if you are interested, you can go to the channel and see the recording of this channel, of this session as well. So once again, thank you so much. Take care. Hopefully we'll meet again.